Ji Assalamu alaikum everyone. How are you all? I hope that you all are doing well. Welcome back to the class. Today we are going to be continuing with the topical MCQs of AS level biology of the biological molecules, and specifically we are discussing the subtopic of testing for the biological molecules. We discussed question 1 to question 6 in our previous part, right? So if you guys want to see from question 1 to question 6, I will be attaching the link of that YouTube video in my description box below. You guys can see that video from there. And today we are going to be beginning with question 7. Also in the previous video I also discussed about, or I delivered the lecture about the test for the starch, test for the reducing sugars, proteins, and the non-reducing sugars in detail. So you guys can go and check that video as well. You will find a detailed lecture of the test for the biological molecules simultaneously with the MCQs as well. So let's begin with question 7. Question number 7 over here is from October November 19, paper 1-3, question 6, right? Alright, so over here basically they are saying that tests for the biological molecules were carried out on a food sample which correctly matches the biological molecules in the food sample with the color for the positive result. We know that starch is a polysaccharide, and for starch we do the iodine test, right? Iodine test, and if the starch is present, the iodine solution turns blue black, right? Fructose, we know that fructose is a reducing sugar, and the test for the reducing sugar is the Benedict solution, and if the fructose is present, then the Benedict solution will change its color from blue to red, orange, yellow, green, whatever, depending upon the concentration of the fructose. Over here it is written red, so we will accept it. Triglycerides are the fats, or you can say the lipids, and it is saying white. So we know that yes, this is also correct because we have studied about the emulsion test or the test for the fats. The test of the fats is basically known as the emulsion test. So what happens over here is that if we have to test for the presence of the fats in a solution, so what we will do is that we are going to add the ethanol to the sample, right? Then we are going to add the water. Wait. First we are going to add the ethanol to the sample, then we are going to add the water, and if the milky white emulsion is produced, so it means that the fat is present. Look, this is a milky white emulsion which is produced. It means that the fat is present in the solution. And if the clear solution is there, like for example, if the whole solution is clear just like this, no white emulsions are present, it means that the fat is absent, right? So I hope that this is clear to all of you now. So similarly over here we were discussing that triglycerides is going to give us the white color, or you can say that milky white precipitates, yeah, milky white emulsion as we discussed over here. So milky white emulsion you can say, not the precipitate. Milky white emulsion is the most appropriate term which is used. So you can say that yes, B is our correct answer over here. Option B is the correct answer. Alright, so now let's move on to question number 8 everyone. Now question number 8 is from May June 19, paper 1-1, question 7. It says that a solution of amylase was added to the suspension of the starch. Alright, so amylase is an enzyme which can digest the starch, or you can say that which can break down the starch, right? And starch is broken down into maltose. Now over here they are saying that after 30 seconds 3 sample of the mixture were tested with the iodine solution, Benedict solution, or with the Biuret reagent. What are the expected results? Alright, so we know that amylase is going to digest the starch into maltose, and we know that maltose is a reducing sugar, right? So starch will obviously, after, okay, they are saying that after 30 seconds, you also have to know that after 30 seconds not all of the starch will vanish. So if we test it with the iodine solution, so iodine solution will give a positive test, right? It will show black color. Why? Blue black color. Why? Because starch would still be present, because they are doing the test after the 30 seconds, which are not enough to fully digest the starch. So starch would be present, and as they are doing it after 30 seconds, so there would be less concentration of the maltose, and less concentration of the maltose means that the Benedict solution will change its color from blue to green, which indicates the lowest concentration of the reducing sugar. Obviously it cannot be red, because red indicates the highest concentration of the reducing sugar, and they are doing the test after the 30 seconds, which means that not all of the starch has been broken down, so hence the not more reducing sugar, for example maltose, is present over there. Biuret reagent indicates the presence of the protein in a solution, right? And if it turns into purple color, and we know that amylase is a protein, right? We know that amylase is an enzyme, and enzyme is a protein in nature, right? So Biuret test will give the purple color, right? So hence the correct answer for this MCQ would be option A. Alright, so now let's move on to question 9 over here. Question 9 over here is from May-June 2019, paper 1-3, question 7. It says that a solution of amylase, which is an enzyme which digests the starch, was suspended to a suspension of starch. The mixture was stirred and kept at 40 degrees Celsius for 45 minutes. If I talk about the ideal conditions for the enzyme amylase, so the ideal condition or the ideal temperature for the enzyme amylase is from 35 to 40 degrees Celsius, right? The samples were then tested with the various reagents. What is the expected set of the result? Alright, so they are saying that there is a suspension of starch, and they are adding the amylase enzyme in it, and amylase can digest the starch and break it down into the reducing sugars, which you can say maltose, right? And we know that maltose is a reducing sugar, right? And maltose, if I talk about maltose, so it is basically made up of the glucose units, right? Now they are saying that what is the expected set of the results when we add the amylase into the suspension of the starch. So then they are saying that the test and the resulting color. If I do the iodine test, we are doing the test after the 45 minutes, right? If I do the iodine test, we know that iodine test is the test for the presence of the starch. After 45 minutes, it is a high possibility that all of the starch may have been digested by the enzyme amylase, right? So there would be no starch present, and if no starch is present, we know that the negative result of the starch is yellow brown suspension, right? So the iodine test will be yellow brown. Then the Benedict test, if I talk about the Benedict test, which is for the reducing sugar, we know that the reducing sugar maltose is present. So the Benedict solution will change its color from blue to orange, right? Then Biuret test, if I talk about the Biuret test, so we know that there is an enzyme which is known as amylase in the solution, and enzymes are protein in nature, right? So protein is present in the suspension, hence so the color change would be purple. The Biuret, you can say that the Biuret solution may change its color from blue to purple, hence the correct answer for this MCQ is option D over here. I hope that this MCQ is clear to all of you. Now let's move on to question 10. Alright, so question 10 over here is from February-March 2019, paper 1 to question 7. It is a very tricky question, no doubt. Now let's analyze it what they have asked us in the question. 4 solutions were tested with the Benedict solution. The table shows the color of the solution after testing. Alright, so basically they have a 4 set of solutions, and they tested each solution with the Benedict solution for the presence of the reducing agents, obviously. Not the reducing agents, the reducing sugars, right? So they have the 4 solutions, and then they have added Benedict solution to it to test the presence of the reducing sugar, and the color change which was observed was this, right? And then they are saying that which row shows the solutions that could have given these results. Alright, so basically then they are saying that solution 1, and then they have given us a value, right? 1 or 0.1% reducing sugar, 0.5% reducing sugar. So we have to identify it by the color change. Obviously we know that Benedict solution is originally blue in color, right? We know this already from our lectures that Benedict solution is originally blue in color, and then whenever the color change happens, it happens to be red, orange, yellow or green. And then the color change basically depends upon the concentration of reducing sugar. The trace amount is represented by green, the low amount is represented by yellow, the moderate by orange, and high by red, right? So basically it is in the order of the increasing concentration of the reducing sugar. If I talk about the values which are given over here, so let's arrange these values from the lowest to the highest, right? If I talk about the lowest value over here, so the lowest value over here is 0.05, then it's 0.5, then it's 1, then it's 0.1. No, this is not the correct order, sorry. 0.1, 0.5 and then 1.0. Yes, this is the correct order. Now we have to arrange it in a way, like for example, if blue is there, so it is clearly indicating that no reducing sugar is present, right? No reducing sugar is present. So you cannot say that 0.5% reducing sugar. No, we will simply eliminate option D. Yes, non-reducing sugar can be there, right? We can say that yes, non-reducing sugar or 0.00% of the reducing sugar is present. We can consider these three options if I talk about the solution 2 only. Then if I talk about the brick red, which is the solution 3, right? It means that the highest concentration of the reducing sugar is present in the solution C. Why? Solution 3. Why? Because it is showing a brick red color, right? So the highest concentration of the reducing sugar in option A is 1, right? You can see 0.051 and 0.101 is the highest one over here. So yes, we can consider option A, that yes, the reducing sugar is present in the highest amount. Then if I talk about option B, so yes, option B also can be considered because it is also showing the reducing sugar in the highest concentration, and then option C can also be considered. Then if I talk about green and yellow, so green, which is of solution 1, the concentration of solution 1 must be lower than the concentration of the solution 4, right? Why? Because solution 1 is green in color, and green is representing a relatively low concentration of the reducing sugar compared to the yellow one, right? Yellow one, the solution which is showing the yellow color, which is the solution 4, may have a higher concentration of the reducing sugar than the solution 1, which is green in color. So basically solution 4 must have a value which is greater than the solution 1. So let's see that which option is correct over here. Solution 4. Alright, if I talk about solution 4, so you can see over here that 0.1 is a higher value than 0.05. So yes, option A over here is our correct answer. If I talk about option B, so you can see over here that 0.1 is less than 0.5. It cannot be possible. Why? Because I have already told you that solution 4 is yellow. It means that it has high concentration of the reducing sugar than the solution 1. If I talk about option C over here, why option C is not correct? Because option C is giving 1% reducing sugar in the solution 1. Now if you guys see over here, so 1% reducing sugar is a very high concentration of the reducing sugar, and with this concentration the solution would have given the brick red or the orange color, not the green color, right? So that's why we are not considering option C as well. The correct answer for this MCQ is option A over here. I hope that this MCQ is clear to all of you. Alright, so now let's move on to question 11. Question 11 is from October-November 2018, paper 1 to question 7. The question says that a sample of solution was tested for the reducing sugar and the result was negative. Right? It means that the reducing sugar was not present because they are saying that the result was negative. Another sample of the same solution was then tested for the non-reducing sugars and the result was positive. Which step in the test for the non-reducing sugar breaks down the glycosidic bond? We know that what are the steps for the non-reducing sugar test. We have to boil it. First we have to boil the solution with the hydrochloric acid, then we have to neutralize it with alkali, and then we have to repeat the Benedict test. Why we have to neutralize it with alkali? Why? Because the Benedict solution works the best in the alkaline conditions. And why do we boil it with the solution, boil the solution with the HCl? Because it is basically a type of hydrolysis which basically breaks down the glycosidic bonds of the non-reducing sugar and convert it into the reducing sugar, so that it may give a positive test with the Benedict solution, right? So basically over here the step in the test for the non-reducing sugar which breaks down the glycosidic bond is the boiling with the hydrochloric acid. So hence the correct answer for this MCQ is option C. Now let's move on to question number 12 over here. This question number 12 is from May June 19 paper 1 to question 5. It says that after boiling a sample of milk with the Benedict solution, a yellow color is observed. Which conclusion about the sample of the milk is correct? Alright, so basically it means that the reducing sugar is present, right? If the Benedict solution is showing the color change from blue to yellow, it means that the reducing sugar is present. Then they are saying that what conclusion about the sample of the milk is correct. We know that milk contains the reducing sugar which is known as the lactose. And if I talk about that, what lactose is made up of, so lactose is basically made up of the glucose plus galactose, right? Glucose plus galactose makes up the lactose. So there is a high concentration of fructose, no. There is a low concentration of sucrose, no, because it is not the milk sugar. Reducing sugars are not present, reducing sugars are present. Hence option B is our correct answer over here, that the reducing sugar is present, and the reducing sugar present in the milk is lactose, which is made up of the glucose and galactose monosaccharides. And how we can say that reducing sugar is present? Because the Benedict solution, which is originally blue in color, is changing its color from blue to yellow, which means that the reducing sugar is present. So I hope that question 12 is clear to all of you. Now let's move on to the last question on the topical MCQs of biological molecules from a part of testing for the biological molecules, right? So from the subtopic testing for biological molecules, this is our last question, question number 13, which we will be doing, right? So let's begin with question number 13. Question number 13 is from October-November 17 paper 1 to question 7. It says that a student carried out 4 tests for the biological molecules on a sample of milk. The test and their results were as follows. Alright, so we have done a question like this above as well. So over here they have given us 4 tests and then they have told us the results of the test as well. Then they are saying that which biological molecule must be present in the milk, and then they have given us the non-reducing sugar, protein, reducing sugar, starch, and we have to tell that out of all of these which biological molecules are basically present in the milk, right? So now let's begin with heating to 80 degrees Celsius with the Benedict solution gave a brick red color. It means that the Benedict solution is changing its color from blue to brick red, which means that the reducing sugar is present. Adding Biuret solution gave the purple color. Biuret solution is originally blue in color, and if it's changing its color to purple, it means that the proteins are present in the solution. Adding iodine solution gave a orange color. We know that the presence of the starch, if the starch is present, then blue black color is observed, but if the starch is absent, then yellow brown color is observed, and it can also go to the shade of orange, so the starch is not present. Boiling with the acid followed by the neutralization, then heating to 80 degrees Celsius with the Benedict solution gave a brick red color. The color change remains the same for the Benedict test, for the non-reducing sugar test as well as the non-reducing sugar test, which means that the non-reducing sugars are also not present, right? If the non-reducing sugar would have been present, and I already told you that when the when the acid hydrolysis is done, the glycosidic bonds are broken down and more reducing sugars would be present in the solution, and a more darker color may be seen, right? A change in the concentration of the reducing sugars must be seen, and then the change in the color must be seen. But as the color change is just the same, so it means that the non-reducing sugars are not present in the solution. Hence the correct answer for this MCQ must be option C, 2 and 3 only, that proteins and reducing sugars are only present in the solution of the milk, right? So I hope that these MCQs are clear to all of you, and each and every test is also clear to all of you. I decided to make a lecture first for the test for non-reducing sugars, and then we solve the MCQ simultaneously so that you guys can get the concepts more clearly. So I really hope that you guys enjoyed solving these past papers, and if you guys did, do consider subscribing this channel and hitting the like button, and do tell me in the comments below that how do you find these lectures, are they useful for you or not. Do remember me in your prayers. Have a nice day. See you guys in the next video with the next part of the topical MCQs of biological molecules. Allah Hafiz everyone.